very much, Tom. And we have Daniel next.
Sorry, what's that?
This is some other slide.
That's brilliant, yes. We took note of that.
Thank you, Nina. Good morning to everyone. My name is Daniel D'Andrea. I am a senior lecturer in data science in the School of Engineering, Mathematics and Technologies. This is a slide just to summarize my research interest. Basically, what I try to do is to integrate different type of data, in general omics data like transcriptomics, genomics, metabolomics, along with the clinical information, and applying a different type of approaches like statistics, bioinformatics in particular, but also machine learning and AI. I try to identify some mechanism affecting complex disease, or try to identify some new methods to be able to analyze this type of complex data, in particular to support researchers to use this data, but also try to identify new experimental directions, in particular for the area of precision medicine. It is what I have done in the last 15 years, and in particular I have applied this type of approach to different type of disease, in particular cancer, but also psychiatric disorders and rare disease. For instance, now we have, in collaboration with the Imperial College, a clinical trial for the first-in-class inhibitor for multiple myeloma and diffuse large B-cell lymphoma. Today I would like to speak for a new project that I'm doing basically I started last year. It is about cancer. When we speak about cancer, you know that we have cancer cells, but the cancer in general is something that it is a very complex environment. So we can have different type of cells with interaction, and this tumor microenvironment is associated with the tumorogenesis and cancer progression. We decided to focus on a specific agglomerate of immunocells that are the TLS, tertiary lymphoid structures, that emerged as a prognostic and predictive biomarker of response to immunotherapy. So by using different type of data, starting from clinical information that we can have for patients to have a snapshot about the tumor's composition and key features, we decided to use also omics technologies, the standard one, that are the bulk sequencing, so analyzing the entire tissue of cancer, but also the single cell sequencing, so we are able to identify the materials in the any specific cells, and also spatial transcriptomics, where we can also identify where the cells are in the tissue. Obviously, individually, these technologies are not able to identify and characterize well the tumor microenvironment, but maybe together we can have a good understanding about what's happening in the tumor microenvironment, and in particular in the TLS. So what we started to do in collaboration with the Italian Institute of Cancer, I work with them just because they already have some patients where they characterized our lung cancer patients, so characterizing poor, good, super good. They have blood adjacent on tumor sites and also tumor sites, and perform an experiment to have different type of omics, spatial transcriptomics, but also single cell omics. We are able to identify these TLS in the tissue and try to identify some dynamics that are able to be associated with the response to immunotherapy and tumor progression. So as you can see, this type of research is a multidisciplinary research. So what I'm looking for, and the input and expertise that I'm looking for, is something that it is related to oncology, medicine, immunology, not only to have patients or experiments, but also some potential new hypotheses that we can test on public available dataset, but also data scientists to identify some new methods to analyze this complex system. That's it. Thank you.